Good morning, students. Hope you are in good health and enjoying the online tutorials well. I regret to miss the face-to-face interaction with my students in my own classroom. Still, with no way out, we wish for a better tomorrow. Here, I would like to praise and thank to the responses I had got from some of you regarding the classes of the previous days, the cell division and the introduction to the chromosomes. Hope that I could satisfy you with my answers well. Today we will discuss about the structure of the chromosomes and the DNA. The major chemical components of chromosomes are nucleic acids, that is the RNA and DNA, and proteins called histones. DNA has negative charges along its length, while histones are positively charged basic protein molecules which are bound to it. The DNA histone complex is called chromatin. DNA forms about 40%, while histone form about 60% of the chromatin matrix. The nucleosome is the fundamental subunit of chromatin. Each nucleosome is composed of a little less than 2 turns of DNA, approximately 146 base pairs, wrapped around a set of 8 histone proteins which are known as a histone octamer. Each histone octamer is composed of 2 copies each of the histone proteins H2A, H2B, H3 and H4 connected by stretches of linker DNA such as H1 and its isoforms. The chain of nucleosomes is then compacted further and forms a highly organized complex of DNA and protein called a chromosome. DNA or deoxyribonucleic acid is found predominantly in the nucleus of cells of plants, animals, prokaryotes and a number of viruses. In eukaryotes, it is associated with protein forming nucleoprotein, whereas in prokaryotes it is not associated with proteins. For example, the bacterial cell. Generally, DNA is double-stranded, but single-stranded is found in some cases such as bacteriophages. DNA is the chemical basis of genes. Genetic codes are encoded in DNA. In nucleic acids, there are 3 major components: the pentose sugar, deoxyribose in DNA, and ribose in RNA. The phosphoric acid and nitrogen bases, which may be purines or pyrimidines. Purines or pyrimidines are heterocyclic aromatic organic compounds which you will be studying in organic chemistry. In nucleic acids, that is DNA and RNA, 3 types of nucleobases are pyrimidine derivatives, namely the cytosine, the thymine and uracil. Purines are the adenine and guanine. The pentose sugar joins each of the 4 nitrogen bases to form 4 different nucleosides. The phosphoric acid joins with the nucleoside of the pentose sugar to form the nucleotides. The nucleotides form the basic unit of nucleic acid. The nucleotides are joined to one another in a chain by covalent bonds between the sugar of one nucleotide and the phosphate of the next, resulting in an alternating sugar-phosphate backbone. The twist or turns in the two sugar phosphate backbones of the double helix occurs every 10 base pairs, which maximizes efficiency of the base pair packing. The sugar in DNA is deoxyribose, which is a pentose or 5 carbon sugar. The sugars are joined together by phosphate groups that form phosphodiester bonds between the third and fifth carbon atoms of adjacent sugar rings. These asymmetric bonds mean a strand of DNA has a direction. In a double helix, the direction of the nucleotides in one strand is opposite to their direction in the other strand. The strands are anti-parallel. The asymmetric ends of DNA strands are said to have a directionality of 5' and 3', with the 5' end having a terminal phosphate group and the 3' end a terminal hydroxyl group. In a DNA double helix, each type of nucleobase on a strand bonds with just one type of nucleobase on the other strand. This is called complementary base pairing. Here, purines form hydrogen bonds to pyrimidine, with adenine bonding only to thymine in 2 hydrogen bonds and cytosine bonding only to guanine in 3 hydrogen bonds. This arrangement of nucleotides binding together across the double helix is called a Watson-Crick base pair. Some prokaryotes, such as bacteria and a few eukaryotes, have extra chromosomal DNA known as plasmids, which are autonomous, self-replicating genetic material. Plasmids have been used extensively in recombinant DNA technology to study gene expression. The genetic material of viruses may be single or double-stranded DNA or RNA. Retroviruses carry their genetic material as single-stranded RNA and produce the enzyme reverse transcriptase, which can generate DNA from the RNA strand. All up to this for today. Study, memorize and note down. And if you have any queries related, feel free to contact. Remain in good health and stay safe at home. Goodbye, students.